Okay, our next speaker this afternoon will be Dr. Peter Gordon, who is a practicing pediatric oncologist here at Minnesota with a clinical focus in leukemia. Dr. Gordon attended the University of Chicago in the medical scientist training program to achieve both the MD and PhD. His work in the laboratory of Dr. Joseph, I was going to check and make sure I got this name right with you, Piccirilli? Okay, Piccirilli. Focused on defining catalytic mechanisms of RNA splitting in ribosomes. After graduation, he pursued residency training in pediatrics at the Children's Hospital Boston and fellowship training in pediatric hematology and oncology at Children's Hospital Boston, as well as at the Dana-Farber Cancer Institute. Dr. Gordon joined the University of Minnesota in 2014 as an assistant professor, where he established a laboratory focused on leukemia research and a clinical practice that also focused on pediatric leukemia patients. Today, he will speak to us on the use of drug-conjugated gold nanoparticles for pediatric cancer therapies. Please join me in welcoming Professor Gordon.
Thank you for the invitation to speak today. I'll just start out by saying I think the idea of this sort of nanomedicine short course is a great one. As I'll describe, when I began the work I'm going to talk about today, I had no background in nanotechnology or really anything even sort of close to nanotechnology. And so I certainly would have benefited a couple years ago having attended this, and certainly I've already learned a lot today. So hopefully it's something that can continue in the future. So, as you sort of alluded to, I'm sort of a pediatric oncologist with interests in clinical leukemia and a lab that really focuses on the sort of leukemia microenvironment and leukemia biology and understanding how extrinsic factors impact the ability of leukemia to grow and survive and become chemo-resistant potentially. And so really it's almost sort of by happenstance that we ended up working on sort of a nanotechnology-based project, and I'll describe that a little bit more as we sort of move into the talk. But because we really sort of approached this from the perspective of pediatric oncology, rather than sort of a long-standing interest in nanotechnology, I want to begin with just 2 or 3 slides on pediatric oncology, because I think it sort of sets the stage for where we were sort of coming from when we began this. Okay, so pediatric cancer, it's the leading non-accidental cause of death in children. There are about 15,000 children each year in the United States who are diagnosed with cancer. Because of improving therapies, there are almost 500,000 survivors of childhood cancer who are currently alive in the United States. And I think actually pediatric oncology is a field where we've really made some tremendous advances in the last couple of decades. And currently our overall cure rates for children with cancer is somewhere approaching 80%, which certainly is a lot different than sort of the adult fields. And within sort of pediatric oncology, there are some real success stories. One of those being acute lymphoblastic leukemia, or ALL. This is the most common pediatric cancer we see. It's about a quarter of all pediatric cancer diagnoses. And you can see that back in the 1960s, it was essentially a universally fatal disease. And today, our cure rates are somewhere approaching 90% for all patients. And for some patients with favorable cytogenetics or other favorable features, we have cure rates of greater than 97%. However, for every success story like ALL, there do remain some challenges in pediatric oncology, and an example of that is sort of the close cousin of ALL, so AML, acute myeloid leukemia, where in AML, despite sort of maximally intensive cytotoxic chemotherapy and in many children stem cell transplant, we're only curing about half of all children with AML. So significant work still remains to be done there. And I think as we're curing more and more children of their cancer, and again, we're getting near 80%, I think we're also coming to appreciate that cure comes at a cost. And this just illustrates that children who are cured of their cancer are living shorter lives than their counterparts. And somewhere greater than 60, probably closer to 80% of children who survive their childhood cancer go on to develop a chronic health condition that can be directly related to the therapy that they received, whether it's chemotherapy or radiation therapy or surgery. And it can affect almost any organ system. And I've listed just a couple of them here, but pretty much if you name an organ system, I can probably think of a toxicity from the therapy that we use to treat their childhood cancer. And since a picture is worth 1,000 words, these are actually identical twins. At the age of 4, the twin on the right was diagnosed with acute lymphoblastic leukemia. She was treated with chemotherapy and cranial radiation. She's clearly now a long-term survivor of her cancer, but I think just this picture illustrates some of the late effects from therapy that she's experienced. So I think with this background in mind, one of the really important themes in pediatric oncology research is not only do we need more efficacious therapies, but we also need therapies that are going to be less toxic, because we want to cure these children of their cancer, and then we want them to go on and live healthy and productive lives. And I've listed here just some of the examples of sort of, I think, novel therapies that have been developed in the last 10 plus years that I think really try to meet this challenge. And so obviously there's molecularly targeted therapy, antibody drug conjugates, sort of the concept of targeting multiple orthogonal cancer pathways, certainly immune and cellular therapies, which are sort of at the forefront of oncology right now, and then obviously nanotechnology. And I've sort of highlighted the first four in black because I think they're things that every week in clinic we are frequently using. Whereas I think it's slightly different. With nanotechnology, while there certainly are some drugs such as PEG asparaginase or liposomal daunorubicin that we use in clinic, I don't think nanotechnology, at least in my experience, has sort of permeated into the pediatric oncology clinic to the same extent as some of these other therapies. And so it's not really something that I'd given a lot of thought to until a couple of years ago when actually I heard a talk, and the speaker in the talk described oligonucleotide functionalized gold nanoparticles that were being used to measure or assess mRNA levels in cells. And this was work that was being developed by Dr. Chad Merkin's lab at Northwestern, and we've heard about some of that already. So I think many of you may be familiar with how it works, but just to sort of run through it for people who aren't. So you start with a gold nanoparticle, you functionalize it with an oligonucleotide, and you make that oligonucleotide sequence complementary to a gene or mRNA that you're interested in within the cell. And then onto this first strand, you anneal on, just via Watson-Crick base pairing, a second shorter oligonucleotide that contains a fluorophore at the end. Now, when this fluorophore conjugated oligonucleotide is sequestered to its complement, the fluorescence is quenched by the gold. However, when this particle is taken up by the cell, the complementary mRNA combined to its complementary sequence on the gold nanoparticle, and in doing so, release the fluorophore conjugated oligonucleotide, and you can measure that fluorescence as a proxy measure for the mRNA levels within the cell. And so when I heard this talk and this technology described, I thought it was really kind of neat and interesting and cool. So I went home and read a little bit more about it, and I sort of discovered that there are some really well-described favorable properties for these particles, including high uptake into diverse cell types. And we've heard about some of these things already today. They're stable in biological environments. They have low toxicity, low immunogenicity. And I think as sort of a non-chemist, non-nanotechnology person, as I was reading the methods of the papers, it actually seemed somewhat straightforward in terms of making these particles. So the starting components were all commercially available. You could buy citrate-capped gold. You could buy oligonucleotides containing a terminal thiol that will attach to the gold. You could buy fluorophore-conjugated oligonucleotides. And then in terms of assembling these particles, it really required sort of a benchtop centrifuge, a pH meter, and a couple of other basic lab equipment. So as I sort of read it, I said, this is actually maybe something that a non-expert could actually try. Clearly, as I read more, there were some challenges, and we've heard about some of these today, including, reticuloendothelial scavenging, the question of how to sort of enhance or optimize endosomal escape once these particles are taken up, and then obviously an issue of sort of background release of the fluorophore-conjugated oligonucleotide from the gold nanoparticle. But I sort of started with sort of a very simple question or idea. What if we replace the fluorophore with a drug? In other words, could we, in effect, generate a gold nanoparticle that selectively releases a drug from sequestration to the particle in the presence of a targeted mRNA that's specific to the cancer cell? So the idea behind this would be that the gold nanoparticle would selectively release the drug only when it encounters the appropriate mRNA, and in doing so would sort of selectively target the cancer cell while sparing a normal cell that lacks that mRNA. So this just sort of looks at it in a little bit more detail. So the idea is we start with a gold, we functionalize it with an oligonucleotide, shown here in red, that's complementary to a sequence within the cancer cell that in theory is unique to the cancer cell. We then anneal on, via Watson-Crick base pairing, a second shorter oligonucleotide that rather than a fluorophore at the end has a drug. These particles are then taken up by cancer cells as well as normal cells in the body. However, when it encounters the cancer cell-specific mRNA, it can bind to its complement on the gold nanoparticle, and in doing so, displace the drug-conjugated oligonucleotide, which can then inhibit its cellular target. And then in a second step, cellular nucleases can actually come along and actually degrade the RNA-DNA hybrid, and in doing so, deplete the cancer cell of an mRNA upon which it may be dependent upon for growth or for survival. And obviously, this is a binomial reaction, so the release of the oligonucleotide conjugated to the drug is going to be dependent both upon the presence as well as the abundance of the targeted mRNA. And so we thought, again, without having done any experiments, that this could be potentially a very customizable approach for activating a drug as well as depleting an mRNA in a cancer cell. And we also thought one of the reasons we were excited about it was that we thought it could be actually applicable for any cancer in which you could sort of identify a drug as well as a unique RNA target. And it's sort of, in my estimation, that pretty much means almost any cancer. You can think of a drug that you would want to deliver to it and an mRNA that's going to be unique to it. So again, things we sort of thought about as we were sort of thinking about should we sort of try this out and see if it works is first is choosing a drug. Obviously, it needs to be clinically relevant. It needs to be chemically amenable to conjugation to DNA, and I think for a chemist that probably means anything can be attached to DNA. For sort of the non-chemist, I wanted to choose something initially that would be relatively straightforward so that I could hopefully convince a chemist that they could do it without too great of an investment of their time and resources. Obviously, the conjugation of the drug to the DNA shouldn't perturb the drug's ability to function. And then thinking about choosing an mRNA. Ideally, it should be an mRNA that the cancer cell is dependent upon for survival, such that you get sort of the synergistic effect that when you deplete the cancer cell of that mRNA, it's going to affect the cancer cell proliferation or survival. Ideally, it could have synergy with the molecularly targeted drug. And then perhaps most importantly, obviously, it should be an mRNA that's unique to a cancer cell. And so you could think about an mRNA or an RNA. It doesn't necessarily have to be an mRNA that's overexpressed. It could be an alternatively spliced mRNA, where the alternative splice junction is going to be unique to the cancer cell. Or, and I'll come back to this, in many cancers, we see translocated genes, where you get a sort of hybrid protein that's made up of protein 1 and protein 2. And certainly at that breakpoint sequence, that sequence is going to be truly unique to the cancer cell. So we wanted to, for proof of principle, we decided to start with a drug called Dasatinib. So many of you may be familiar with it. It is a multi-kinase inhibitor. We use it in leukemia therapy, in particular in a disease called CML. We're also testing it in solid tumors and a variety of other cancers. It targets a number of kinases, including SERC, LIN, BCR-ABL, which is the mutation that we see in CML, as well as c-KIT. It has a hydroxyl group, which makes it very amenable to conjugation, again, for the non-chemist. There was also some previous literature suggesting you could modify this hydroxyl group, and Dasatinib was still able to bind to its targets. So that was certainly another reason we thought this would be an interesting drug to start with. And then finally, it has because it's a kinase targeting drug, there are some very well-established drug activities and assays and targets that we could use to sort of characterize. Again, we viewed this as proof of principle and not as sort of the appropriate therapeutic drug for this technology. And the reason for that is, as many of you may know, Dasatinib is an orally taken drug. It's generally well tolerated with sort of mild to moderate side effects. But again, we thought for these reasons, it made sense to start with in terms of proof of principle. So the first step, obviously, is conjugating Dasatinib to an oligonucleotide. And for this, I turned to Nansheng Li and Joe Piccirilli's lab at the University of Chicago. And so he converted the hydroxyl group to an azide, which could then be reacted with an oligonucleotide that terminates in an alkyne group using click chemistry. This reaction occurred with good yield and good purity. And the other advantage, I think, of this technology is that terminal alkyne oligonucleotides are commercially available. And so I could order a whole slew of oligonucleotides of different sequences, all terminating in an alkyne group. And then in sort of one fell swoop, we could react it with our Dasatinib azide to generate a variety of different Dasatinib conjugated oligonucleotides of varying sequences. So the first step we then wanted to ask was, does conjugating Dasatinib to an oligonucleotide perturb its efficacy? Because obviously, if we're screwing up its efficacy, it's no longer going to be sort of useful. So the first thing we used was a SERC kinase activity. As I mentioned, Dasatinib inhibits SERC, and so we used a SERC luciferase assay, added either free Dasatinib or our Dasatinib DNA oligonucleotide, and you can see the IC50s are essentially the same. We then generated gold nanoparticles with our dasatinib DNA oligonucleotides and added them to the same SERC enzyme. And you can see that for the first couple of conditions, SERC retains full activity, suggesting that the dasatinib oligonucleotide does effectively remain sequestered to the gold nanoparticle and cannot sort of bind to its target SERC. However, in the last condition, we added an additional oligonucleotide to the reaction that basically mimicked the mRNA in the cell that our gold nanoparticles were targeting. And what you can see now is we see complete inhibition of SERC activity, suggesting that that oligonucleotide was able to displace the dasatinib conjugated oligonucleotide from the gold nanoparticle, and then it could bind to and inhibit SERC. So that was certainly encouraging that, again, conjugating dasatinib to an oligonucleotide didn't perturb its efficacy. We then wanted to ask the simple question of do leukemia cells, because that's what we were going to first test in it, actually take up gold nanoparticles. So the first step is we just generated gold nanoparticles that were labeled with a fluorophore. Then we incubated them with a whole variety of human leukemia cells, some ALL, some AML. The next day we did flow cytometry and just looked at the percent of cells that were positive for gold nanoparticles. And you can see that essentially 100% of the cells after an overnight incubation had taken up the gold nanoparticles. Now, to make it a little bit more realistic and sort of mimic what's going to be happening in vivo and in the bone marrow, we then took our same leukemia cells and incubated them with an excess of human bone marrow cells or murine bone marrow cells, going up to a sort of 1,000 or 100-fold excess of the normal cells, added our fluorophore-labeled gold particles, let them incubate overnight, and then again the next day looked by flow cytometry. And you can see again that these leukemia cells very efficiently took up the gold nanoparticles even in the presence of an excess of normal cells. And then finally, we wanted to ask, is this also going to hold true in vivo? And so we actually generated mice that had subcutaneous leukemia tumors. Then we did a single intratumoral injection of fluorophore-labeled gold nanoparticles. And then the next day, we euthanized the mouse, excised the tumor, digested the tumor, and looked by flow cytometry. And you can see that about 75% of the leukemia cells within the tumor had taken up gold nanoparticles after a single injection into the tumor. So all of this data suggested that leukemia probably is a reasonable model to sort of start with for testing these particles. So then we actually wanted to select an mRNA target, and we decided to start with the survivin gene for a number of reasons. First, it's also called BRC5, and so on some slides it may be listed as BRC5. It's an inhibitor of apoptosis, so knocking out survivin could be beneficial. It's highly expressed in most tumors, including leukemia, but is absent or low in most differentiated cells. So first, we wanted to show sort of specificity of our particles for the survivin gene. So we actually generated a murine cell line that expresses human survivin mRNA under the control of doxycycline. So if we add doxycycline to the media, survivin mRNA expression is turned on. If we take the doxycycline away, the mRNA expression turns off. We then generated gold nanoparticles targeting the survivin sequence, but again, rather than dasatinib, we included a fluorophore. And so again, when it's sequestered to the gold nanoparticle, its fluorescence is quenched, but if it's displaced by the binding of the survivin mRNA, it now fluoresces. So then we added equal amounts of these gold nanoparticles to our engineered cell line in either the absence or presence of doxycycline. And what you can see in the absence of doxycycline and the absence of survivin mRNA, we see relatively low levels of fluorescence indicative of sort of background release of our fluorophore conjugated oligonucleotide. However, in the presence of doxycycline and in the presence of survivin mRNA, we see a significant increase in fluorescence, again indicative of release of the fluorophore conjugated oligonucleotide from its sequestration to the gold nanoparticle. So at this point, we then actually wanted to ask, can these things actually target what we think they should be targeting in the leukemia cells? And so we first wanted to look at two enzymes within leukemia cells that we know are targets of dasatinib. So these are SERC, again, we saw this enzyme earlier, as well as CRKL. And so what we did is we incubated human leukemia cells with either dasatinib, in which the sequence specificity was scrambled, dasatinib by itself, or our dasatinib gold nanoparticles targeting survivin. And what you can see is that dasatinib by itself inhibits both phospho-SERC as well as phospho-CRKL, which again is sort of what we would expect in leukemia cells. But we also see that our dasatinib DNA gold nanoparticles also effectively inhibit SERC, phospho-SERC, and phospho-CRKL, whereas in contrast, our dasatinib gold nanoparticles with a scrambled sequence, where we basically took the survivin sequence, scrambled it, have very minimal effect on both phospho-Circ as well as phospho-CRKL signaling. So this was certainly encouraging. And as I showed you on earlier slides, there's also data from Dr. Merkin's lab and others that as sort of a second step, cellular nucleases can come along and degrade the RNA-DNA hybrid. And so we wanted to ask, do we also deplete the survivin mRNA in the cells that have taken up the gold nanoparticles? So we added either gold nanoparticles in which the sequence is scrambled, gold nanoparticles in which they were targeting the survivin gene. And what you can see is we get about a 20% knockdown in the survivin levels when it's targeting survivin as compared to the scrambled gold nanoparticles. And actually, if we change the backbone of the DNA to a locked nucleic acid backbone, and again, this was described in the literature, we get even better knockdown, closer to about 60%. So again, this work sort of suggested that our particles were behaving as we'd hoped. So we then wanted to ask, do they have any therapeutic efficacy in the treatment of leukemia? So for this experiment, we started with 3 cell lines, so 3 leukemia cell lines that are known to be sensitive to dasatinib. So Kasumi and SKN01 are AML cell lines that have activating KIT mutations, and c-KIT is an enzyme that's inhibited by dasatinib. K562 is a CML leukemia cell line that has a BCR-ABL translocation that's inhibited by dasatinib. And finally, as a control, we use 32D cells, which is a mouse hematopoietic cell line that is largely dasatinib insensitive. We then treated our leukemia cells in vitro with either gold nanoparticles containing DNA targeting survivin or gold nanoparticles targeting survivin conjugated to dasatinib. And you can see that in the cell lines known to be dasatinib sensitive, we significantly decreased their viability with the dasatinib DNA gold nanoparticles, but only really have a minimal effect with the gold nanoparticles that lack dasatinib. And in contrast, in our control cell line that's largely dasatinib insensitive, we see very little effect. So we then went ahead and did a dose titration, and this just illustrates that we start to see an effect. So here we're measuring annexin positivity as a measure of apoptosis, that really at about 150 picomoles, we start to see efficacy of these gold nanoparticles. We then went on to finally show that the dasatinib gold nanoparticles can actually prolong the survival of mice that are transplanted with human leukemia cells that are known to be dasatinib sensitive. So shown in green are mice that were treated with our dasatinib gold nanoparticles. You can see their lifespan was over doubled in this experiment, whereas gold nanoparticles targeting survivin but lacking dasatinib really only had a minimal and not statistically significant effect on the survival of the mice. So I think these experiments in our mind really kind of showed proof of principle for this approach. But as I sort of alluded to earlier, dasatinib is an oral drug. Patients take it at home. Again, it's generally well tolerated. So therapeutically, it doesn't make sense to attach it to a gold nanoparticle. We're basically taking it and making it much more complicated than it needs to be. And so we started to think about sort of, well, what are other more relevant drugs that we could turn to? But before we did that, we actually wanted to ask, do dasatinib gold nanoparticles exhibit less toxicity than dasatinib itself against cells that don't express the targeted mRNA? So for these experiments, we turned to 2 different types of cells that are known to be inhibited or affected by dasatinib. The first are human hematopoietic stem cells, which are also called CD34 cells, and then T cell activation. And what you can see is that dasatinib inhibits phospho-Circ in human CD34 cells, and this is known to affect their survival and proliferation ability. In contrast, our dasatinib DNA gold nanoparticles really have no effect on these cells that, again, have low levels of survivin. Similarly, if we treat human T cells with CD3, CD28 beads, we can essentially turn them on or stimulate them. This can be blocked by dasatinib. However, again, our dasatinib gold nanoparticles really only have a small effect in terms of blocking T cell activation. Again, suggesting that in cells that have lower survivin mRNA levels, these particles are going to be less toxic than against the cancer cells that have higher levels. However, as I've alluded to sort of earlier, is that it seems in our hands that many normal cells seem to take up these particles less well than leukemia cells and cancer cells. And at least that may be sort of confounding these toxicity experiments. And this is shown here. If we take human hematopoietic stem cells again, or leukemia cells, incubate them with equal amounts of fluorescent gold nanoparticles, and then look at their uptake the next day, you can see that our leukemia cells have taken up significantly more, again on a log scale, particles than the human hematopoietic stem cells. Similarly, if we take human T cells, what you can see is that T cells, so CD8 subset of T cells, actually take up the particles quite poorly, whereas in contrast, the CD4 T cells take up the particles quite well. And so again, this is just illustrating that different cells take up these particles with different affinities. So in order to sort of address this issue in terms of our specificity experiments, because again, this could really be sort of confounding how we're thinking about things, we actually turned back to our cell line that we engineered to express human survivin mRNA. And so we then incubated those cells in the presence or absence of doxycycline, treated the cells with either Dasatinib or our Dasatinib gold nanoparticles targeting survivin, and then again looked at phospho-Circ by flow cytometry. And what you can see is that for our Dasatinib DNA gold nanoparticles, only in the presence of DOX do we see inhibition of phospho-Circ, again suggesting that the Dasatinib conjugated oligonucleotide does in fact remain sequestered to the gold nanoparticle in the absence of survivin. So this sort of leads us to sort of have a model for the diminished toxicity being sort of twofold. First, there's decreased uptake of gold nanoparticles by many normal cells. Obviously, there are other cells, in the liver and reticuloendothelial system that may take them up very well. But I think as a general principle, we found that many of sort of the normal cells seem to take them up less well. And then second of all, there's decreased release of the drug from the gold nanoparticles in the normal cells that lack the targeted mRNA. So again, at this point, we really wanted to then sort of shift gears and actually turn to what we think would be a more therapeutically relevant drug. So something we would actually potentially use this technology with. And so for this, we turned to a drug called SN38. It's the active metabolite of irinotecan. So irinotecan is a drug that we actually use frequently in clinic for a number of different tumors. And SN38 is about 100 to 1,000-fold more potent. Its potency in part is determined by sort of the liver activation of the drug, which again is going to depend upon the patient. It's a topoisomerase 1 inhibitor. SN38 itself isn't given clinically due to poor solubility and toxicity. There is a sort of wide body of literature suggesting it's amenable to conjugation and modification, particularly at the C20 hydroxyl. And again, it was a drug that we thought could be modified relatively easily for non-chemists. So again, our idea was relatively simple. Basically, we would generate our same gold nanoparticles, but rather than having a fluorophore Dasatinib conjugated to the oligonucleotide, we would have SN38. It would be taken up by cells. When encountering its complementary mRNA, it would bind to it, to the gold nanoparticle, and displace the SN38 oligonucleotide, which could then bind to topoisomerase 1. So again, the first step was conjugating SN38 to an oligonucleotide. And we took a very similar approach, except this was done by John Wyden and Dan Harkey's lab here at the U. And again, converted the hydroxyl group to an azide, which we could then react with commercially available oligonucleotides that have a terminal alkyne. Again, relatively good purity, relatively good yield, relatively short synthesis for them. So the first thing we then wanted to, again, ask was, does conjugating SN38 to an oligonucleotide perturb its efficacy? Again, there was some literature to suggest that it wouldn't, but we first just added our SN38 DNA construct to a number of cells that are known to be SN38 sensitive, and we got it into the cells using lipofectamine, and what you can see is that these cells all exhibited sensitivity to the SN38 DNA construct, again suggesting that the conjugation itself didn't necessarily inhibit the ability of SN38 to target topoisomerase. We then decided to sort of test our gold nanoparticles in the context of a cancer called Ewing sarcoma. The reasons we chose it, it's irinotecan sensitive, which again is sort of the precursor to SN38. It's the second most common bone malignancy that we see. About half of patients are adolescents or teenagers. We treat them with very intensive chemotherapy, surgery, radiation therapy. Survival is only 70% for localized disease, which isn't great. And for metastatic disease, it's about 25%, which is really terrible. And as I sort of alluded to, therapy is very, very intensive. And so these children have significant both acute and late toxicities. And so I think it's a disease where we certainly need to make an improvement, and it's a disease where we know there is some sensitivity to irinotecan. So the first thing we wanted to ask again is, do Ewing sarcoma cells take up gold nanoparticles? So again, we just generated gold nanoparticles containing a fluorescent label. We incubated them with 4 different Ewing sarcoma cell lines. The next day, we looked by flow cytometry, and you can see that the cells, about 100% of the cells for all of these cell lines had taken up gold nanoparticles by the next day. It is somewhat time dependent, where within a couple of hours, we're already seeing uptake, and then sort of by the next day, it's sort of maxed out. So again, we wanted to sort of more accurately mimic what's going to be happening in vivo. So we took our Ewing sarcoma cell lines and we mixed them with increasing numbers of normal bone marrow nuclear cells, again up to 1,000-fold excess, added our fluorescently labeled gold nanoparticles, and then again looked at uptake the next day. And again, what you can see is that essentially 100% of the Ewing sarcoma cells took up gold nanoparticles even when outnumbered 1,000-fold by normal bone marrow cells. So again, suggesting this is probably a tumor where this technology might work. And then finally, we generated mice that had subcutaneous flank tumors comprised of Ewing sarcoma cells. We then did a single intratumor injection of fluorescently labeled gold nanoparticles. The next day, we euthanized the mouse, resected the tumors, and looked at what % of the tumor cells had taken up gold nanoparticles. You can see again, after a single injection, about 65% of the Ewing sarcoma cells had in fact taken up gold nanoparticles. So we then wanted to ask, well, what mRNA should we target in Ewing sarcoma? And we decided to start with the EWS fly gene. And so in about 85% of Ewing sarcoma tumors, there's a translocation involving chromosome 11 and 22, and it generates the EWS fly protein. And what you can see here is that when you go from the EWS sequence to the fly1 sequence, this sequence here, where you go from gene 1 to gene 2, is going to be truly unique to the cancer cell. So no normal cells in the body should have this sequence. And so it seemed like a good target for this technology. And the other reason we thought this would be potentially a good place to start is because translocations are common features of many malignancies, and many malignancies are dependent upon the translocations for survival. And I've listed just a few of the different cancers here with some of the translocations that we see. And again, our idea would be that our drug-conjugated oligonucleotide would essentially span the breakpoint of the translocation as you go from sequence 1 to sequence 2, in effect. So again, to sort of test the specificity, we generated a cell line, or engineered a cell line that expresses EWS fly1 mRNA under the control doxycycline. And then we added gold nanoparticles that were fluorescently labeled targeting EWS fly1 in either the absence or presence of doxycycline. Again, in the absence of doxycycline, we see just sort of background levels of fluorescence and release, but that's significantly increased in the presence of doxycycline and in the presence of the EWS fly1 mRNA. The other target we decided to look at was going back to the survivin gene. As I mentioned, this is sort of ubiquitous across many cancers. And this is just a Western blot showing a number of different Ewing sarcoma cell lines, and they all express survivin in fairly high levels. Additionally, this was another paper out there that actually showed that patients who had high survivin levels in Ewing sarcoma actually did significantly worse than patients with low levels, again, suggesting it was a good target for Ewing sarcoma. So we then went ahead and generated gold nanoparticles containing SN38 conjugated oligonucleotides. We first just characterized how many SN38 oligonucleotides were bound to each gold nanoparticle, and there are about 40 per gold nanoparticle, which is sort of agrees with what is described often in the literature. We then added our SN38 gold nanoparticles to Ewing sarcoma cells in vitro and just looked at viability. And we added either SN38 scrambled gold nanoparticles, in which this targeting sequence was scrambled, or particles that were targeting the survivin gene or the EWS fly1 translocation. Again, you can see that for the 4 EWS fly1 positive Ewing sarcoma cell lines, both the survivin targeting and the EWS fly targeting nanoparticles significantly reduced the viability in vitro. As a control, we used U2OS cells. So this is actually an osteosarcoma, so a different type of bone tumor cell line. It lacks an EWS fly1 translocation, but does express survivin. And sort of reassuringly, what you can see is that the survivin gold nanoparticles did decrease the viability of the U2OS cells, but the EWS fly nanoparticles had essentially no effect on the U2OS cells, which is what we'd expect, again, because they do not express EWS fly1. So we then again did a dose titration, and again, what you can see in terms of viability is that at sort of sub-nanomolar concentrations, we start to see an effect on viability. So relatively low doses of these nanoparticles can start to really target these Ewing sarcoma cells. We then wanted to ask, could SN38 gold nanoparticles inhibit colony formation in vitro? And this is sort of a proxy assay that you can do in vitro that looks at sort of the tumor forming ability of individual cells. And so in effect, in these colony forming assays, you're asking, how many individual Ewing sarcoma cells can go on to sort of recapitulate a tumor? So you can see with the absence of gold nanoparticles, after about 10 to 14 days of growth, you can see quite a few particles, which again we quantitated over here. If we add SN38 gold nanoparticles in which we scramble the target sequence, we again see essentially equivalent or close to equivalent number of colonies. However, if we add gold nanoparticles that target the survivin gene, we see significantly diminished number of colonies. And similarly, if we add gold nanoparticles targeting with SN38 that target EWS-FLI1, again, we see a significant reduction in colonies, again, suggesting that these can effectively target Ewing sarcoma cells in vitro and prevent their ability to proliferate and sort of regenerate a tumor in vitro. And then finally, we wanted to ask, do these have any efficacy in vivo? So again, we generated mice that had subcutaneous flank tumors comprised of Ewing sarcoma cells. We did a single intratumoral injection of either survivin gold nanoparticles or survivin gold nanoparticles with SN38. The next, 48 hours later, we excised the tumor, dissociated it, and looked at tumor viability. And you can see that the survivin gold nanoparticles conjugated to SN38 diminished the tumor viability by about 60% after a single intratumoral injection. So again, providing at least some in vivo data suggesting this could be an effective therapy. So just to sort of wrap things up, we think this has the potential to be a customizable approach to activate a drug as well as potentially mRNA depletion in cancer cells. Obviously, most of our experiments in vivo have been done with intratumoral injections just because scaling has been sort of an issue. Obviously, really they should and need to be done with IV injections. That's a place we'd like to go. Obviously, we'd like to extend to other cancers, other drugs, other mRNA targets. I will say we've tried some other mRNA targets, and some seem to work better than others. And that's sort of been our limited experience, that maybe every mRNA target is not going to work well. And there is some trial and error, at least in our hands. Obviously, we'd like to optimize mRNA knockdown, since, as I showed you, it's somewhat modest under sort of baseline conditions, as well as address some of the other challenges in the field, such as enhancing endosomal escape, the question of background release of the drug-conjugated oligonucleotide, reticular endothelial scavenging, and as well as others. So just going to conclude by thanking the people who did it and funding. The work with the Dasatinib was mainly done by Nathan Gasai, who's a pediatric oncology fellow in the lab. And then Jordan Nauman, who is a technician, did most of the work with SN38. The initial funding for the Dasatinib came from Leukemia Lymphoma Society New Idea Award. And then follow-up work with SN38 was supported by the Children's Cancer Research Fund. Thanks, everybody, and I'm happy to take questions. Yeah, the question is, inhibiting one mRNA or targeting one mRNA may not be enough, and have we thought about targeting multiple? And the answer to that is yes. One of the nice things about this technology is that it's very easy to functionalize the gold nanoparticles with any sequence that you want, and in fact, you can do a mixture of sequences. So you could generate a single gold nanoparticle that has sequences that recognize more than one mRNA. So in theory, and we've thought about this, we just haven't done it, we could generate a gold nanoparticle, a single gold nanoparticle that targets both survivin and EWS-FLI1, or whatever sort of genes or mRNAs of interest that you'd want to target. And again, you're absolutely right, that may be more effective than sort of targeting a single mRNA. Yeah, so the question is sort of thinking about sort of cancer cells that can have very high levels of transcription. Have we looked at pharmacokinetics? And the answer is sort of that we really haven't. In theory, our idea was that the more mRNA, in theory, the more release of the drug-conjugated particle. And I would say in general, we've found that to be sort of largely true, but it's not always a perfect correlation. And I think that at some point you do sort of saturate the release of the drug-conjugated oligonucleotide. So, in the sort of doxycycline experiments, we can, at some levels, control the levels of mRNA, and so we can see sort of more effect given in the presence of more mRNA. But in vivo, it hasn't always been sort of that straightforward, I guess I would say. So I think that probably is an area that also needs some more investigation. And similarly, sort of along that path, we certainly see background release in the absence of mRNA. And I think that's sort of inherent to this assay. The reality is these are drugs that we're giving to patients anyways, and so a little bit of background release isn't necessarily a problem, but it is something that we do see. Yeah, that's a great question. So the question is, how much nanoparticle actually gets into the tumor itself, how much actually gets taken up by the tumor cells, and sort of clinically, sort of how do we think about that? And I think you sort of hit on sort of one of the points I sort of made earlier is that I don't think nanotechnology, beyond sort of like pegylating some of our drugs, has really made it necessarily to the pediatric oncology clinic. And I think part of the reason is what you get at, that I think there still are issues with getting enough into tumor cells and getting uptake and systemic injection, and how do you bypass scavenging by the liver. And so I think those are all problems and issues that are sort of being addressed by the field by sort of people who are more experienced and more knowledgeable about nanotechnology than certainly I am. But I think those are all sort of limitations to the technology, and I think some of them should be able to be overcome. But certainly I think right now it is a limitation. Certainly there are papers from Dr. Merkin's group and other groups showing that you can systemically inject gold nanoparticles, and in fact they do get into tumors. Again, it takes a lot. You really have to scale up if you're going to be giving these through the tail vein. And so we've sort of gotten by doing intratumoral injections, since it requires a lot less. Certainly in the clinic there are cancers where you could envision doing intratumoral injections. So, do you have to do everything systemic? Not necessarily, but certainly I think ideally that would be the case.
Well, very good. Thank you very much.
Thank you.